Okay, so I'll be talking on emerging diversity of DNA structure. DNA is a molecule which is close to my heart. And why only my heart? I think, and DNA is close to everybody's heart. And, I have been teaching this course using a textbook. And that textbook is written by Richard Sinden. He's at A&M University, Texas. So, every book is dedicated in their own way. So this was interesting. So I'm sharing it with you. So Professor Sinden dedicates this book in these words. He says that this is dedicated to my parents for sharing their DNA. And then, yeah, I'll just narrate this. Just a minute. Yeah, dedicated to my parents for sharing the DNA, and dedicated to my wife for sharing her DNA with me, and dedicated to my children, David and Laura. May you treat the DNA of your ancestors with love, respect, and care. So this itself, this dedication itself tells you that how important is this molecule, and it is within all, and it is the master molecule, I think, which is controlling all the organisms or all the life in the on the earth. Before I start, let me just clear that the contents of this talk are prepared for educational purpose only. Contents are collected from published textbooks, magazines, journals, and other sources. Okay, so if we talk about thinking, the thinking process, the questioning process, this is started from Socrates. He says, I cannot teach anybody anything. I can only make them think. And here is the crux. If you start thinking, you will start analyzing, and then you can make this world a happy place by your analytical power, thinking power, and then creativity. At the same time, Richard Feynman is a very famous. He was a physicist, and he worked on quantum mechanics and dynamics. But at the same time, there is already a volume of his famous lectures. He has been a great orator. So what he says that the internal machinery of life, the chemistry of the parts, is something beautiful. And it turns out that all life is interconnected with all other life. And this is true. And see, if there is a term life, DNA has to be there. This is an important thing to mention here. So 6 decades ago, more than 6 decades ago, I think, scientists proved that our destiny is not written on the forehead, but it is beautifully coded in the string of 3 letters words of DNA molecule, the blueprint of life. A long helical ladder tightly coiled for it to be housed in the tiny nucleus of our body cells. We all know this. So I'll give you some salient features. So the DNA is more than a pretty molecule called an instruction manual. It contains the information needed to construct and operate the human body. And let me tell you that DNA is like a computer program, but far, far more advanced. than any software ever created. And these have been words by words of Bill Gates, the chairman of Microsoft. Surprisingly, there is 2.5 meter of DNA in each of our cells, and that is about 3 billion bases, a base pairs, you can say. So about 98% of the human DNA has no function, and it is called junk DNA. This is so surprising. Okay, in fact, this talks about evolution, that how with time few of the genes were became silent and all that. So in 2003 November issue of Scientific American published an article, and it is Gem in the Junk, in which it stated that the failure to recognize the importance of introns may well go down as one of the biggest mistakes in the history of molecular biology. A study shines a new light on molecular tools of our cells used to govern, regulate gene expression. There are about 25,000 genes in a human genome. If you see the magnitude of the genome, it seems really surprising that there are only 25% or 25,000 genes present there. So human DNA is 99% identical to that of chimpanzee. Okay, but between human, DNA varies only by 0.2%. So you and me, we just vary by 0.2 and 0.1%. So all human beings differ from one another. So in physical appearance, susceptibility to disease, susceptibility to disease, and response to medication. That one, I am affected more by one drug and the other is not. So all these changes are due to variation in DNA. This variation is called single nucleotide polymorphism. So SNPs, frequently occurring genetic variants, are those. And these are single nucleotide polymorphism, we call it. So great importance of small difference. And here are the examples. See, one base difference may change the color of flowers on the same branch. At the same time, one base difference may change the texture of the hair. Just one base change. That is why we call it single nucleotide polymorphism. Fine. So of the 3.2 billion bases, roughly 99.9% are the same between two people. I told you, right? It is the variation in the remaining tiny fraction of the genome, 0.1%, roughly several million bases, that makes the person unique. This is really amazing. This small amount of variation determines attributes such as how a person looks or disease or the disease he develops. Oh, this is so surprising. Can you believe that Neanderthal man DNA makes 1 to 4% of our genome? Okay, you'll be surprised to see this. Now, using DNA analysis, scientists are illuminating our evolutionary history. This is only possible through DNA. Recent findings shed light on Neanderthals and ourselves, our past and our future, by locating which genes are different in human and Neanderthals. It may be possible to discover why our species survived and that or there went extinct. This is an example of a modern man. So Dr. Pabo's research team reconstructed the Neanderthal genome from skeletal remains and compared it to the 5 modern people of the varying ancestry, including Chinese, Guinea, South Africa, West African, and French people. all except Africans shared Neanderthal DNA. Clear? So the construction of Neanderthal genome changed our views on the origin of species. The controversial story is that modern man and Neanderthal split from a common ancestor about 400,000 years ago in Africa, which with Neanderthals moving north to dwell in Europe until they became extinct about 30,000 years ago. So this is the evolutionary aspect. But at the same time, another aspect of DNA, a trip to gym can alter your DNA. This is surprising. Okay, and this is already reported. So what it says, bout of exercise sees methyl groups removed from metabolic genes. So if your metabolic genes are methylated, it means they are transcriptionally inactivated. But if you go for exercise, they are these methyl groups are removed, and then gene will be transcriptionally active. This is epigenetics, right? So it is the study of changes in gene expression based on mechanisms other than changes in the genetic code itself. One such epigenetic mechanism is methylation. Okay, so most of so if you are a chemist, you will really understand this. The bonding of methyl groups to C5 location of the cytosine. Cytosines are methylated. Okay, C5 location of cytosine based in the DNA strand. Epigenetic modifications are heritable. Clear? So these are the heritable changes in chromosomal structure that modify gene expression without altering the DNA sequence. While genetic mutations are permanent, epigenetic modifications are potentially reversible. Okay? So it depends in which environment the organism is growing. This is very, very interesting, and this relates we all. Have you heard of Eve DNA? There is Adam and there is Eve. If I talk about Eve DNA, it has to be something from the female part. Okay, so our precious Eve DNA. Why I'm calling it precious? Because the mitochondrial genome is passed down from the mother. Okay, so what more precious can be than our mother? So whatever energy you have, whatever energy I have, that has come from the mitochondria, which were part of the egg, right? The sperms could not give their mitochondria to the egg due to technical reasons. I'll tell you that. Okay, so whatever we have and whatever we are here, that comes from our mother. When we talk about mitochondria, once when mitochondria was there, it was as a free cell, right? It was a prokaryote and it was a free cell, and then there came a theory. That theory is symbiotic theory. It says that endosymbiosis is the process where one organism lives inside other, and it works out really well for everyone. That is what happens with our mitochondria. So what happened that mitochondria, we can say invaded, or maybe we can say that the eukaryotic cell that engulfed mitochondria, this free living prokaryote once upon a time, and then there was a good understanding, we can say, and then it stayed inside the eukaryote only. So mitochondrial DNA, it looks like, since it was a prokaryote, so the mitochondrial DNA was circular, like we have plasmid DNA. Okay, so the mammalian mitochondrial genome is transcribed exclusively through the female germline. So the human mitochondrial DNA, which we call mtDNA, is a double-stranded circular molecule of 16,569 base pairs, and it only contains 37 genes, coding for 2 RNAs, rRNAs, 22 tRNAs, and 13 polypeptides, means 13 proteins. So these 30, out of these 37 genes, so they are giving 13 polypeptides. And these proteins are used by the mitochondria, especially for its greatest function, and that is electron transport chain. So the complexes are there which are mediated by enzymes. Those enzymes come from these 13 proteins. So they swap their genes among themselves. Genes are essential for the normal mitochondrial function. Enzymes involved in oxidative phosphorylation. Okay, this is a part of electron transport chain. So I'll tell you what happens actually. This is the sperm. And the mitochondria is this, the spiral mitochondria is here. So it takes a spiral shape. It is, most of the energy is used in the travel through fallopian tubes and all. So what happens, that the middle piece is called power of the sperm because it has to move, and the numerous mitochondria coiling around axial filament produce energy for the movement of the tail. So once it reaches there, the sperm mitochondria is destroyed after implanting in an egg. So all your mitochondrial DNA comes from your mom's egg. Clear? Okay. So this was one of the interesting fact about DNA, which I wanted to tell you. So when we talk about genome, so we will also include, in case of plants, we will also include chloroplast DNA, we will include mitochondrial DNA and the nucleate DNA. But in case of animals, so it will be mitochondrial DNA and the nucleate DNA that makes the complete genome of any organism. This is a simple human karyotype. And why I'm showing you this, we already know that we have 23 pair of chromosomes, and this is a picture, we call it human karyotype. Interesting thing is this, see this 23rd pair, it is X and Y. Okay, so if it is XY, it's a boy. If it is XX, it's a female. You all know this. See the size of X and see the size of Y, right? So this seems to be scary, maybe if it keeps on reducing its size like this with evolution, Y may not survive. Maybe, on a lighter side, maybe this is the reason that these X traits are so dominant, right? And you can also say that they have to, their cell has to work more, and more genes have to be expressed. They have to carry much more, or they have to do much more than a male organism. So maybe the size difference could reflect in all these functions. Anyway, this was just the lighter side. Okay, fine. Now I'm a chemist. Now I will look DNA at a different angle. So a chemist's visualization about this molecule of life is, it's engineering, basically bond engineering, and then manipulating DNA for any function we have in mind. So it's the endless march of alphabets in a 4-letter alphabet, but you don't see that until you open it, the helix, and look inside. Okay, so a structural biologist or a chemist would do that. There are two views of a DNA. Maybe these audience are mainly from zoology or biochemistry or biotechnology background. So there is one view, that is biochemist view. Biochemist only talks about the overall shape, ignores atoms and bonds. He doesn't have to do anything with atoms and bonds. But the chemist talks about atom by atom assembly and functional groups. Chemical nature of nucleic acids, you all know it. I'll just pass through it. Surprisingly, two kinds of phosphoesters are there in nature. When we talk about ester, see, whenever there is a term ester, two things come in mind, an alcohol and an acid. So alcohol plus acid, they make an ester. Clear? So there are two kinds of phosphoesters. Now, phosphoric acid also can be an acid to make an ester. So two kinds of phosphoesters are present in nature. Chicoic acid, they are phosphoesters of phosphoric acid and ribitol or glycerol. So the OH group comes from glycerol or ribitol. So they are linked by phosphodiester bonds. These acids can be found in the cell wall of gram-positive bacteria. The main function of teichoic acid is to provide rigidity to the cell wall. And here is the example, So this is ribitol, and then these are, the phosphoric acid is here, and then this kind of polymer is formed. The ribitol teichoic acid, we'll call it. Okay, it's not, if it is a ribose ester, then that will be there in our nucleotides. So nucleic acids are phosphoesters. So I told you teichoic acids are there, which are there in cell wall, and then nucleic acids, which are there in all mammals and all. So nucleic acids are phosphoesters of the phosphoric acid and ribose. Like teichoic acid, they also make backbone of the phosphodiester bonds. They are present in cell nucleus or cytoplasm. Being the fundamental substance of all the living organisms, nucleic acids derive their name from the nuclein word, which was discovered by Friedrich Mischer in 1869. You might know this. So there are two kinds of nucleic acids, RNA and DNA. Clear? So a section of DNA looks like this. If we talk about the chemical constituents, you all know it. There is a base. Bases could be purines, pyrimidines. Then there are sugars and phosphoric acid. Okay, so sugar and phosphoric acid will be linked, and then they form an ester bond. Now let me tell you, this beautiful double helix model, this has come not just like this in 1953, but not only in 1953. The discoveries have been there long back. So the people contributed toward discovery of the DNA structure were Levin and Jacob in 1909 to 1929. So what they did, they talked about the structure of nucleotide. They talked about the link, that the sugar is linked to base, and the base is also linked to phosphate. Through enzymatic and acidic hydrolysis, they did it. Then Takahashi gave a tetranucleotide model that A, G, C, T are there in equal concentration, and then they are linked together. Fine. And then came Esbury. He gave the first X-ray diffraction pattern of dried DNA fiber, but the base spacing, what he found was 3.4 angstrom. Clear? So that gave the first idea that there is base sparing and there is something ladder-like kind of thing, which has the base pairs and the spacing is 3.4 angstrom. Then a very important discovery. Gulen, he gave the concept of hydrogen bonding through viscosity. What he found that if we add nucleotides in water, they form viscous solution. So it can only be viscous if there is a phenomenon of hydrogen bonding there. So for that, enolic forms have to be there. So enolic forms of bases were then discovered through Gulen's discovery. Then came Alexander Todd in 1944. So he synthesized the first nucleotide in his laboratory. But then there has been a galaxy of people. Erwin Chargaff, you know what he did. Maurice Wilkins, Rosalind Franklin, Francis Crick, James Watson, and Linus Pauling all have contributed in their own way. We cannot, undermine anybody's contribution here. But the lucky people were three: Maurice Wilkins, Francis Crick, and James Watson. Okay, and they got the Nobel Prize in 1962. There is another story why Rosalind Franklin was not involved, was not part of this. Maybe it is not given posthumously, and she died at the age of 37. Many things. Linus Pauling also did a DNA triple helix structure, but it was not proved later on. So, but everybody, Erwin Chargaff gave a wonderful theory that purine and pyrimidine will be there in 1:1 ratio. So all these facts were there, and then finally Wilkins, Francis, and James Watson came up with the model. Now, important thing. So this is DNA. It is double. DNA is double. This is a wonderful information, and it really revolutionized whole of the molecular biology. And it is a helix. That is a geometrical consequence. If you have a thread, and if you just leave it on the table, the thread will be in helical shape on its own, a cotton thread. But if you stretch it, it will become a linear molecule. Similarly, if it is a helix, it is a geometrical consequence of many things. So DNA is double and helix. Important thing is that it is double and it is helix. This gave a great aspect, a great concept of base pairing. So base pairing is defining property of DNA and was particularly exciting at it suggested the copying mechanism of DNA. This was the idea of Francis Crick, which they have mentioned in their seminal paper of DNA discovery, DNA double helix in 1953. So it is double and helix. So nature has done an incredible design, and you have to be amazed and amused all the time. I do all the time. I keep thinking, for a small thing to make, for any small molecule, organic molecule to make in the laboratory, we have to have an infrastructure. See how beautifully nature has designed its molecules, and the molecules which are really at the heart of the life. Then came this lady, we have to recognize. So the model which Watson and Crick gave, it was a beautiful DNA double helix model. But a great concept, an idea which was given by Rosalind Franklin, was that DNA is structurally polymorphic. So in 1952, see, this is before DNA double helix was deciphered. 1952, Franklin took the best DNA X-ray diffraction pictures. And people say that she worked so much, the 100 hours of exposure to the X-ray only brought her at this stage that she had an ovarian cancer. Okay, anyway, so Franklin distinguished between two types of DNA crystals. According to the condition, it could be a dry DNA and it could be a wet DNA. So dry form is different and wet form is different. Later on, the A DNA was the dry DNA and B DNA was the wet form, which is the part of our cell. Clear? Other, the same, purines, pyrimidines, but there is a reason why I'm showing you this. The reason is that if you see the structure, you will see that all the 9 atoms of the this heteronuclear base, all the 9 atoms are in one plane. Similarly, in the pyrimidines, all the 6 planes atoms of this helix, not helix, ring. All the ring 9 atoms and all the ring 6 atoms are on the same plane. Means you cannot fold it. It is planar because it is aromatic. Clear? But this is not the case with deoxyribose and ribose. So ribose rings are puckered. Okay, so they are not rigid. They are not planar. So what can be there? DNA, in case of DNA, this deoxyribose, 2 deoxyribose. So at one time, this 1 and 2, this could be above the plane. Above the plane means towards C2H5OH. So this is this condition. We call it C2 endo conformation. If it comes down, it will be C2 exo conformation. So sugars are not planar like bases. So it could be above the plane and below the plane. Fine. At the same time, this like the C2, the C3 carbon can also go up the plane. So if it goes up the plane, you will have C3 endo conformation and C3 exo conformation. What is found in nature? That all the DNA molecules have C2 endo conformation mainly, and all the RNA molecules have C3 endo conformation. RNA can never be C2 endo conformation. RNA is, so this is one of the difference when we talk about what are the difference between DNA and RNA. So one of the difference is this, that the sugar puckering is different. In DNA, it is C2 endo conformation, and in RNA, it is C3 endo conformation, right? And the consequence of this is this. See, if it is C2 endo conformation, the distance between this phosphate and this phosphate will be 7 angstrom. But at the same time, if it is C3 endo conformation, the phosphate distance is 5.9 angstrom. This is going to have a great consequence when we talk about the total DNA double helix or the various forms of double helix. Clear? Similarly, if O is above the plane towards C2H5OH, then it is, we call it O1 endo. Okay, O4 endo conformation, and then O4 exo conformation if it is below. But then you see 4.6 angstrom and 2.9 angstrom. So these small, these intricate differences, small differences in DNA, in the constituents of DNA, especially sugar, can have a big impact. Finally, so we have sugar, we have phosphate, we have nitrogenous base, and the link is like this. This is known as nucleotide. You all know it. I'll not elaborate on it. Now, this nitrogenous base, which is linked to this C1, we call it glycosidic linkage. We call it N-glycosidic linkage. Clear? Now, this N-glycosidic linkage, if we turn it 180 degree, it will be this form. So we say it's syn form or anti-form. Anti-form is that when this ring, the larger ring is away from the sugar. When it is above the sugar, above this oxygen, it is a syn form. It is analogous to cis and trans kind of conformation we have. Why I'm telling you all this? Because all these small parameters will have a great impact on the overall structure of DNA and its polymorphic forms. So DNA is a double strand, double helix with 2 strand base paired by hydrogen bonds. You know this, okay? You know this that there are two phosphodiester backbones. One is this, another is this, and then there is also a difference in polarity. I'll just tell you that. Now, we just talked about the base pairing. If A base pairs to T and G base pairs to C, now this base pairing is not exactly like this. This base pairing have these have different differences. You know, if it is a long axis, there can be roll. You know, the bases sometimes can roll upon other. Okay, then there can be slight. Fine. If at the same time there can be a tilt. You know, one base pair is this, other base pair is this. So it is not exactly same. It can be like this. It can be shift also. So rolling, sliding, tilting, shifting, right? These are small parameters, and then twisting, okay, and then rise. So there is a book by we call it Bible of these nucleic acid structure, by Frederick Sanger. So if you get a chance, you can see all these details there. All these differences, they give up to structural polymorphism. What is structural polymorphism? So polymorphism refers to the existence of DNA in various forms. It depends on the base composition and on physical conditions. Means, like Rosalind Franklin said about wet DNA and the dry DNA, means conditions were different. They differed in humidity. On the basis of that, they categorized DNA molecules on aDNA, bDNA, and zDNA, which have different parameters, and there is a difference in that. So major polymorphic forms, though native form of the DNA is the pDNA, I already told you, it is the wet form, but depending on the humidity, it can exist in different forms. pDNA, you already know. How do they differ? bDNA is 92% humidity, which is there in cytoplasm. Then aDNA is 79% humidity. zDNA is 60% humidity, means if you keep on getting it dried, the crystals, they will have different structures. So aDNA is like this and zDNA is like this. zDNA, when you talk about zDNA, you know, it differs in its helical sense. It becomes left-handed. aDNA, bDNA, both are right-handed helices, but zDNA becomes left-handed. Minute differences are there, so I'll not talk more about it, but I'll tell you why do we call it zDNA. Okay, we call it zDNA because you see the glycosidic bond. If you talk about glycosidic bond, see in case of aDNA, it is anti-anti. bDNA, it is anti-anti, but it is in case of zDNA, it is anti and syn. At the same time, there is C2 endo conformation and there is C3 endo conformation. So if you take all these things together, anti-syn, anti-syn, C2 endo, C3 endo, C3 endo, C3 endo, this gives a zigzag shape. And due to that, we call it zDNA, right? This is important to tell you. So it is not clear, you know, when we talk about polymorphism, this is really not a story which will ever end. So it is now clear that the DNA molecule can assume different structures depending on the base sequence and environment. So in addition to classic B form structure, several other forms have been observed. Among them are A, B, Z DNA. They are identified as the major forms, you know it. But a detailed inspection of the literature reveals that only the letter F, Q, U, V, and Y are now available to describe any new DNA structure that may appear in the future and will hopefully complete the glossary of DNA structures from A to Z. Okay. Professor Manju Bansal from ISC Bangalore, she has written a beautiful article about it. And some of these contents are from her article. Factors stabilizing double helix. This is also important to talk about. You know, this molecule, which is there from the time when Neanderthal people were, living. So it has to be a very, very stable molecule. What are the factors that stabilize DNA over RNA or DNA double helix? Simple, they are simple physical chemistry rules. Hydrogen bonding, we all know it. I will not elaborate on it. Base stacking. Base stacking, since the aromatic bases are planar, I already told you, so they can stack nicely on one another. This tendency of bases is a consequence of the attempt of bases to minimize the contact with water. I told you that, just for an example, if you have got anthracene with you, and if you put an anthracene is a tricyclic hydrocarbon. If you put it in water, you will have clumps of anthracene. Okay, you will have clumps because anthracene molecules they aggregate, and they aggregate because one sits above other. So similarly, and that is stacking interaction, we call it. At the end, the van der Waals interactions are also there. So existing between base pairs. Similarly, the base pairs also would like to, G would like to go above G and C would like to go above C. So, so these are termed as base stacking, which stabilizes DNA. This is one of the important regions. Electrostatic interaction. We already know this. So electrostatic interaction, DNA is present in the vicinity of salt. See, if you put DNA in a deionized solution, do you know what will happen? Both the strands will separate. Why? Because there is repulsion. Both the phosphodiester bonds, both the phosphodiester chains, which are negatively charged due to phosphoric acid, due to phosphate, they are negatively charged. So there will be repulsion. For that, what nature has done, it is there in the soup. It is there in the soup, nucleoplasm, or if RNA is there in the cytoplasm. So there are already these salts. There is magnesium, there is sodium, there is potassium. So they partially neutralize the phosphodiester backbone, the negative charge, and then they remain intact. So this is one of the reasons. Similarly, to take a sample of DNA, if you put some alcohol in it, you know what will happen? The DNA will precipitate. You know why? Because the water will be taken up by the alcohol. And then DNA will not have any water around it. It will precipitate. Similarly, and this method is used for the isolation of plasmid. Last में absolute alcohol डालते हैं तो उससे DNA precipitate हो जाता है. Why? Because there is a hydration spine. See, this is guanine, this is cytosine. So both are hydrogen bonded. But you see, in the major groove and in minor groove, there is the hydrogen bonded with water. So if this water is not there, what will happen? It will precipitate. It will aggregate. Okay, but this does not disturb the double helix structure. Let me tell you, it will become condensed, but it will not denature. Okay. DNA denaturation, I just told you why. Why do we call it denaturation? Because the hydrogen bonding pattern will be disturbed. Clear. So this is, if this is DNA double helix, if you give it a high temperature because of beautiful complementarity, and since with energy these hydrogen bonds will be disturbed, so DNA strands will be separated. So we call it denaturation because it is not the natural state of DNA. But then there can be renaturation. This is a wonderful property of DNA, that it can denature and it can renature. This is the only region that you can still isolate DNA of our ancestors, and from the from 200,000 back. If there was any generation, or if there was any organism, if we have the fossil, you check out the DNA, and then we can still work on it, because it got dried up. It got dried up, but then it was not destroyed that way. Fine. So it can again be renatured. How? If you give it a proper condition. So slowly cool or lower the pH, or give put it in a buffer, it will again form DNA double helix, because salts are also important for stabilization purpose. Let me tell you, none of the biological molecule, not only DNA, none of the biological molecule work alone. And this is the same with society also. You cannot survive alone. It's very, very difficult. We have to interact. We have to interact. Even if you are in a class, you have to interact with your colleagues, with your lab mates, or with your classmates. If you are teachers, you have to have an association. What I mean to say that since we are social animals, we have to interact. But this is an analogy. This is nothing to do with this particular slide. So let me tell you, DNA doesn't work on its own. It means it has to interact. It is going to be a very, very passive molecule if I put it outside the cell. It will not work on its own if it is not interacting with proteins, with cofactors, with salts, and if it is not stabilized. This is a beautiful example. During DNA replication, you will come across all these enzymes which help DNA in opening up, and then which help DNA in being there. When DNA opens up during replication, one can think that if it is opening up, it can again go back, because there is self-complementarity. Two strands are complementary, but then there are proteins, single-strand binding proteins. These single-strand binding proteins will bind on the single strands which have arisen there, and then they will not let the helix again to go back. Right? Once replication is over, then they are off from the site. And what I mean to say that any biological phenomenon which is mediated, any biochemical reaction which is being mediated by enzymes or other factors, they have to interact with the biomolecule. Either it is protein, or it is lipid, or it is carbohydrate, or it is your DNA. So the enzyme helicase, it recognizes origin of replication, and it causes unwinding of DNA. Because if DNA has to open up, it has to unwind. So for unwinding, there are particular enzymes which will open up it. And helicases are those enzymes which will do this job. Okay, single strand binding proteins are there, I just told you. Then two separate strands act as templates. Okay, binding of DNA is followed by synthesis of RNA primers. You already know this. So it's all goes into action. Now, if there is any structure, it has a meaning. Nature has not done anything without a purpose. So if it is DNA is double helix and it has got grooves, it has a particular reason. So majors and minor grooves are there. You see, major grooves, minor groove. But how do they look like? So major grooves, if you see the major groove side, it is there. If you see the minor groove side, you see that this N7 portion, N7 side is the major groove side, and this other side is the minor groove side. So there are clear-cut defined. So we also said it is present in the N7 of the purine. Minor grooves are the N7, N3 side of the purine. So these two groups provide very distinct surfaces. which proteins can interact. So these groups are not just like that. They are there for the interaction. I just told you that DNA cannot work on its own. So certain chemicals and drugs, they can interact specifically with either major group or the minor group. This is the same thing, there is base pairing, and then it talks about the. So this is the overall nice, detailed structure of a double strand. So sugar phosphate backbones are there. Important thing to tell you is here that both the strands have different polarity. This goes to 3' to 5', and this goes to 5' to 3'. Clear? And that depends on the places where phosphate is attached. If it is attached to the C5, it will be C5 portion, and if it is the 3', this is going to be 3'. Okay. I think most of you know this. Now, I'll also tell you something about how and why this DNA interaction is important. So it is a target for many drugs under clinical trials. The modulation of gene expression and protein synthesis, activation and inhibition of DNA function. What I'm talking about is that the significance of interactions, why DNA would interact with foreign molecules. To know mechanism of the interaction is also important, and development of drugs for better efficacy. This is the importance of DNA drug interactions. There are going to be two kinds of interactions: non-covalent, covalent interaction. So non-covalent interaction can be intercalation. Intercalation means if there is a base pair, any planar molecule will go and sit inside the base pair. So it could be also, so the molecule can also go to minor group. And if it is a covalent interaction, there can be conjugation with proteins. That is mainly done in the major group. Significance of groups. See, this slide clearly tells you that intercalation is this. Then you have got major group binding, okay, and you have got minor group binding. There are molecules, the chemists have synthesized molecules which would be specific to the major group and minor group. So this particular, Hooks 33258, this goes to, Hooks goes to minor group mainly, okay. Okay, minor group binding, neomycin also goes to minor group. Now I'll tell you, there is interaction, there is proflavin. Proflavin molecule, since it is a planar molecule, it will interact inside. Fuchs is also minor group binder, but I think I don't know why is it showing major group binding here. But anyway, similarly, there are different molecules which differ in their side chains, but then the ligand will interact and it will be intercalation kind of thing. Similarly, proteins can bind to DNA through bases, sugars, and the phosphate groups. Hydrogen atoms, hydrogen bonds, phosphate are not specific, because phosphate will interact non-specifically because of its electrostatic nature. But the great importance of stabilizing protein DNA interaction. So any positively charged protein can interact with DNA, which is negatively charged, and then a protein DNA complex there. And the greatest example is histone examples that gives the way to the formation of nucleosomes and all this, so that this long DNA double helix is coiled in a very, very small nucleus, which is tightly packed there. So guanine exposes greater number of potential hydrogen bonding sites. I'll tell you that. Then the polar charged residues of amino acids play a crucial role, like arginine is there, lysine, serine, threonine. Okay, they all interact. Acidic residues are used sparingly. aspartic acid and glutamic acid, then only glycine makes a significant number of interactions, okay, being very, very small. So few interactions are produced by hydrophobic residues. So I'll just go through it. Favored amino acid based hydrogen bonds are there. Arginine, lysine, they interact specifically to guanine, aspartic acid and glutamic acid to adenine, serine and histidine to guanine, and 80% of the serine and threonine interaction are with the DNA backbone. Hydrogen bonding geometry, it could be single, it could be bidentate, it could be complex. Okay, so if you have protein DNA interaction, you can bend the DNA. Okay, but this will be there just through protein DNA interactions. Two possible reasons for bending could be special base sequences or structural motifs are repeated in DNA helix. Sequence-specific bending by the proteins. Here is nucleosome. See, I just gave you an example of nucleosome. This is nothing but a beautiful example of DNA-protein interaction. Okay, so you see a long DNA strand, double helix, which is then bound to histone proteins, and these histone proteins are lying here. Okay, they are positively charged. They have positively charged amino acids like lysine, arginine, and all. So this is the way that the hierarchy is there, and DNA condense. So initially it is a 2 nanometer, then it becomes 10 nanometer, 10 nanometer, then 30 nanometer, and the coiling, super coiling, then 300 nanometer, 700 nanometer, finally 1400 nanometer. This width is attained by the chromosomes. But this, the basis of all this is nothing but the DNA protein interactions. I already told you about it. No, this is important to tell you, the stability, why DNA is more stable than RNA. So you see RNA has an OH group here. So this is, I mean, it will attack on phosphate, and then what will happen? It may cleave it. So it will cleave it, and then you will have this kind of this uses in alkaline medium this ribose 2 hydroxyl group not present in DNA. So DNA is not going to have this kind of cleavages. So that is why there is more stable molecule. Okay, so we call it base catalyzed hydrolysis of RNA. Now, this is important. Various hydrogen bonding pattern schemes, and they will give you higher order DNA structure in solution. We know Watson-Crick double helix. It's a very simple G-C hydrogen bonding. Now, what happens if you just rotate it or shift it 180 degree shifting, it will give you reverse Watson-Crick. So what will happen? In Watson-Crick base pairing, you have two strands, and though the two strands are anti-parallel to each other, what will happen if you reverse it? It's going to give you a parallel DNA double helix. So there is a possibility that there can be parallel DNA double helix. Okay, certainly based on conditions, but this is one possibility which gives us, which is given through this hydrogen bonding. Then there is Hogestyn. Hogestyn in 1963 gave another idea. What he thought that there are still potential hydrogen bonding sites on guanine and adenine which can hydrogen bond. See, okay, so here these sites are being used. These sites are being used. And then if you again put it 180 degree, it will give you reverse Hogestyn hydrogen bonding. So various hydrogen bonding patterns can be generated. So DNA bases have such an potential. There is another remarkable form of the base doublets. So you see, AT is a simple hydrogen bonding, A to T, G to C. You see, there are still hydrogen bonding sites. So another base can come and sit on it. This is Watson-Crick, this is Watson-Crick. But if you take about a Augustine hydrogen bonding, it can go here. So now you have AT, which was already there in the duplex, and then T. So you have TAT. You have C plus. A cytosine has to be protonated for this NH. So you have C plus GC. Clear? We call it Augustine hydrogen bonding. And then similarly, AT and GC. So this still can accommodate, A can accommodate A, G can accommodate G. So it is not that always A will pair with T. Sometimes, given the condition, A can also pair with A, G can also pair with G. This is in reverse Augustine. So in one molecule, in one structure, you can have Augustine hydrogen bonding and Watson-Crick hydrogen bonding at the same time. But then this has great implications. Formation of base triplets. So doublet is ATGC, but then there are triplets. Base triplets can be there. And then these triplets will give you great structures. So jab we met means another DNA molecule, whether it is single strand or double strand, it will come and make you this. So this, I already told you, this is the DNA duplex, but this has space where a third strand can nicely accommodate and form this. See how nicely it goes, and it is not disturbing the DNA double helix. Again, do it. You see, we call it triplex forming nucleotide. See how nicely it goes and sits there, and it has great significance. These were first observed by in poly A and poly U and Folsenfield and Alexander Rich, 1957. But then its biological significance, the potential biological function of these triple helices was postulated in 1968 when Morgan and Wells reported that triplex formation was able to inhibit transcription. Similarly, in these, it is not that we always have to have a third strand to make it. Intramolecule, there can be intermolecule and intramolecule. So Claude Lenin, with whom I worked for some time in 90s, he has been a great biophysical and biophysicist. Okay, so he worked on small DNA triple helices. Similarly, you don't have to have a third strand, so intramolecular DNA triple helices also will be there. Means if you have a duplex, there will be some condition that the third strand will come out and it will form a duplex. Okay, so Frank Kaminsky in 1987 gave this feature. But there is prerequisite. Not every strand and not every segment of DNA can form a triple helix. There has to be a polypurine and polypurine stretch. As you hear. So there has to be polypurine and there has to be polypyrimidine stretch. And only then a third strand can go and sit here. So the TFO means the third strand should either be polypurine and polypyrimidine. And the hydrogen bonding's characters will be the same which I explained where I talked about these triplets. So we, in my laboratory, we have worked on all these DNA triple helices. We have shown that this can be there. So I tell you, what is the significance of this targeting of the duplex? So if you want to target a DNA duplex, either you fix this duplex, you have to have a major group or minor group binding molecules which will go and bind there specifically, or you have to have something where the strand would would bind more specifically to one of the strand, and one strand will be out. Okay, and these such strands are, such nucleic acids are known as peptide nucleic acid, because the backbone is the peptide. And then I already told you the third, if the third strand can form a triplex using all those strategies, you can block a particular gene. And central dogma of molecular biology, you already know this. The DNA gives you mRNA, and that gives you protein. So oligonucleotide directed triplex formation leads to very interesting and novel applications. What is that? If you have a DNA duplex, you can form a third strand, I already told you, and then it will give you a triplex. Now, if it is a gene, and if it forms a triplex, it will not be transcribed. No transcription. So if this is a disease-causing protein, disease-causing gene, you can stop this gene, and you can stop inhibition of transcription. We call it gene strategy. Okay, these all strategies are being worked out. They have their limitations, but people are still working on it. At the same time, there are triplex helix, triplex specific ligands. These ligands stabilize the helix, and then transcription can be halted. Similarly, if you this is DNA, and you know that as a result of transcription, it will give you mRNA. Then inhibition of translation can take place if you make an antisense which is complementary to this. Then no protein formation will take place. This is antisense strategies. Okay, the concept, this is more in practice, I think. The concept behind antisense oligonucleotide gene silencing or antisense therapy was introduced in 1978. Okay, and they came up with the idea after they used the antisense oligonucleotides to stop viral replication in cell culture. So first used was flower saver, a genetically modified tomato, as the first commercial grown-up genetically engineered food to be granted license from human consumption. So all these have been done using our antisense technology. I'll not go into detail, but antisense RNA technology is also used for the cancer therapy. This therapy is used for treating brain cancer, malignant glioma, and cancer of prostate glands. These all reports are there. So I just wanted to tell you the importance of these. Similarly, FDA has approved some of these drugs, which are antisense oligonucleotides, and they have also shown their potential. I'll tell you about one more molecule, one more base, and that is guanine. Guanine is again a very wonderful molecule. Okay, so out of the 5 nucleotides commonly found in DNA and RNA, only guanosine is capable of forming extensive self-structures in solution via Augustine hydrogen bonding with it to give GG base pair. So G base pairs with C is not a big thing, it is always there. But due to its potential hydrogen bonding sites, G also can hydrogen bond with G. Like this. If you put 4 Gs like this, you will have a Chaturbhuj kind of thing, which has got 4 arms. So each guanine has 4 arms now. 2 arms here, 2 arms here. But this is only possible if it is kept in an aqueous solution. So guanine will form gels. Clear? And it forms, due to this hydrogen bonding potential, it forms gels in aqueous solutions. So overall structure of G quartet is stabilized by, but this is nicely stabilized by monovalent cations, and more appropriately by potassium, because it fits in the it nicely fits in the cavity. Okay, so this M, which is monovalent cation, this fits in the cavity. Clear? So DNA quadruplexes play a key role in capping and protecting the ends of the chromosomes, as well as regulating gene expressions. Okay, so what happens if you have got a long G stretch? You will have you will have this kind of structures, okay, which looks like they look like shoe case and all. Now, if it folds back, it can also, here 1, 2, 3, 4, 4 strands were needed to form a tetramolecular structure. Now, if you have 2 strands, but if they are folded, you can have a bimolecular structure. There, so 4-stranded DNA structure, the parallel quadruplexes, all the strands are parallel, and then it will form anti-parallel quadruplexes because when they combine, the polarity will be changed. It's the same thing. So why I showed you this, because if this is the DNA, if this is the chromosome and if this is the DNA, we see that the end of the chromosomes, the 3' overhang is a single-stranded one. We see that these portions are known as telomeres, and they have long G stretches. So they are basically repeated Gs. So in the case of mammals, or especially mainly the eukaryotes, it is T2AG3. So there will be hundreds of repeats of T2AG3. So if it is a T2AG3 here, this might fold, and this might protect the chromosome. Okay, and then we have already shown this. This kind of studies are being done in my lab. So we have already shown that these antiparallel quadruplexes can be formed. Then we have given a postulation that during cell division, antiparallel portions can come and they can form hydrogen bonds, and this kind of hydrogen bonding is done. Okay, so biological significance, if it is a long DNA sequence, and if there are guanines, it can fold back and form this, and then these can be, if normally transcription is activated, if these structures are there, if these structures are not recognized by transcription factors, so transcription can stop. So these are the ligands, again, synthetic chemists have synthesized, which will go and bind especially to these structure-specific quadruplexes. And then, so how a DNA can go and how a ligand can go and bind? It can go and bind like this. Okay, so it can go and stack between the quartets, or it can do the stacking. Okay, so similarly, guanine is also very, very susceptible for oxidation. So if guanine is oxidized, during oxidative stress, so it forms 8-oxoguanine. So in that case, guanine will not do its original work. So if in this case, if transcription is on, and if it is oxidized, this guanine forms 8-oxoguanine due to free radical scavenging. Then what will happen? Formation of free radicals, and due to oxidative stress, transcription can be off. But then there are methods in the cell where a DNA can be repaired. So after repairing, this can again go on. So what I mean to say that this guanine, which is susceptible to oxidative stress, and with the reaction with the hydroxyl radicals, it can be damaged. And we have recently published this, and there's a nice review which only talks about guanine tracks and genomic locations. So the possible biological model, I already gave it to you. Similarly, if there is cytosine, and if it is a low pH, cytosine can also give you CC base pair. So it is not that cytosine will always go with G. Cytosine can also go with C, C+. Okay, so tetramolecular, this way, if we have got 2 cytosine strands, it can give you 2 intercalated motifs. Okay, so we call it I motif because 2 strands which are parallel and hydrogen bonded, they then become anti-parallel, and there is intercalation. So we call it I motif or intercalating motif. So what I wanted to tell you that these nucleic acid bases, which look very simple, they can really give you a galaxy of DNA structures. And if in one of the DNA segment you have G portion and you have C portion, there is a possibility that you have both these structures. Now, these structures, there are reports that these structures are recognized by specific proteins, and then they may give some biological implications. So, structure can assume various forms depending on the sequence. So, it is this feature that ultimately determines DNA structural forms. So, our DNA in cells does not exhibit only one form, but it exists in different forms throughout the length of the DNA. Any change in one position, it will transmit in all along this. And DNA shows a variety of structural features like single-strand DNA, double-strand DNA, triple-strand DNA, four-strand DNA, curved or bent DNA, king DNA, hairpin or cruciforms, relaxed DNA, supercoiled forms, and perhaps the other forms. So it depends upon the sequence context, its association with other components, concentration of water, pH, temperature, and ionic concentrations, which give a galaxy of polymorphic DNA sequences. So this is, if there are repeats, they will give you, according to the repeats, they will give you different structures, triplex, Z-DNA, cruciform DNA, bent DNA, Z-tetraplex, C-tetraplex. So these are all sequences specific. So if you, I told you as a result of tilting, sliding, DNA can give you a variety of structures, and I also call it DNA choreography. So as a consequence of bending, twisting, tilting, rolling features of the bases, sugars, you have a carnival of DNA secondary structures. So all these structures are possible. In the middle, you have our most celebrated form of DNA, and that is canonical B-DNA structure. So but there are possibilities, and people keep manipulating DNA according to their conditions and according to their need. But if a DNA is curved, it has to interact with proteins. So protein DNA interaction, protein ligand DNA ligand interaction, drug interaction also becomes a very important topic. So we have we had a review where we gave this picture, and which it really looks like a bouquet of DNA structures, and it talks about the emerging diversity. So I'll just, in 1 minute, I'll just tell you that my research here interests DNA structural polymorphism, multi-stranded DNA structures, DNA drug-protein interactions, molecular crowding. Okay, these are some of the some of the papers from the lab where we talked about hairpin DNA switching, cruciform switching, and then hairpin duplex equilibrium, which have some biological significance to human beta globin gene cluster. Then we mainly work with structural polymorphism, and we gave various models its role in biology and all that. So then we also talk about presence of divalent cation, which is sometimes mandatory for purine motif triplex, where the third strand is purine. But in special conditions, we say that it's not mandatory all the time. Then short GC palindromic structures. See, just a small example. These are two small oligonucleotides, and you see GGCC and it is CCGG. So this GGCC it gives you two structures in CD spectrum. But the CCGG gives you only one structure. So just a small variation at a small segment can really modify the DNA structure. Then we also talked about the triple-stranded DNA quadruplexes. DNA can, you don't need only 4 strands to form a quadruplex. There can be 2 strands or 3 strands. Then motif structures are there. We nicely show that with concentration or with variation of the structure, this one duplex can form another bulge duplex kind of things and all. Okay, then we have also used ligands to stabilize the DNA quadruplex. But then we also found out that some ligands will destabilize DNA quadruplex, which are located near human myosin gene. So these wonderful advances would not have been possible without the knowledge of DNA structure. In Infinite Nature's Infinite Book of Secrecy, a little I can read. Yeah, it is true that there is so much to know, so much to learn, that this life is really short to explore everything which is there in nature and to relish this importance. But we have to be grateful to God for our creation, that we were here and we are able to, especially the human being, which has a cognition power, and we can really think and analyze all the things around. So thank you so much for your patience. It has been really long.